I'm Hannah Harvey, and today I'm going to talk about one major way our bodies make energy, through the Krebs cycle. Before you get overwhelmed just at the idea of learning a bunch of tongue-twisting scientific vocabulary, let's first think about the big picture. The Krebs cycle, also known as the tricarboxylic acid cycle, the TCA cycle, or the citric acid cycle, involves a ton of teeny tiny reactions that are happening in your teeny tiny cells even as we speak. What's happening in your cells may seem insignificant or small, but added together, these reactions affect your whole body. Your daily life requires energy, namely food. Once digested carbohydrates, fats, and proteins are broken down into their simplest building blocks, enzymes in the mitochondria of our cells further break down these chemicals to form energy, or ATP. For use in the TCA cycle, your food breaks down into pyruvate, fatty acids, and amino acids, which are then turned into acetyl-CoA. To make things simpler, let's just focus on metabolizing carbohydrates. Glucose is first broken down through glycolysis to create pyruvate. In the mitochondria, the pyruvate dehydrogenase enzyme complex turns pyruvate into acetyl-CoA plus an NADH molecule. In order for the Krebs cycle to start, we also need oxaloacetate. The Krebs cycle is called a cycle in large part because oxaloacetate plays a part in starting the cycle, and oxaloacetate is an end product of the cycle. So oxaloacetate is ready and waiting once your food is broken down into acetyl-CoA to start turning that fuel into energy. Acetyl-CoA and oxaloacetate form citrate with the help of an enzyme called citrate synthase. The citrate molecule is then reconfigured into isocitrate by an enzyme called aconitase. Then another enzyme gets involved, named isocitrate dehydrogenase. Not surprisingly, this enzyme dehydrogenates isocitrate to form alpha-ketoglutarate along with carbon dioxide and NADH. Then alpha-ketoglutarate dehydrogenase comes along to change alpha-ketoglutarate into succinyl-CoA, carbon dioxide, and NADH. Succinyl-CoA then becomes succinate with the enzyme succinyl-CoA synthetase, and here ATP is formed. But we're not quite done. Remember, we still need to make more oxaloacetate so that the cycle can begin again. So succinate is turned into fumarate by succinate dehydrogenase, then into malate by fumarase. And finally, oxaloacetate and NADH are made from malate by malate dehydrogenase, and oxaloacetate is now available to start another cycle with incoming acetyl-CoA. Thanks to the Krebs cycle, you can now run faster and jump higher. Enjoy your energy!
Created using Powtoon.